Particularly, I work on the hepatitis B leading to the liver cancer. Really, these patients are not only the hepatitis B influence, it's a non-alcoholic steatohepatitis also, and alcoholic disease-induced HCC also. So this talk is. So I am from the Institute of Liver and Biliary Sciences. This is the super specialty hospital in New Delhi, and this is one of the best in Asia because we are leaders in the cirrhotic and the HCC patient care. And this institute has the dedicated research and of course along with the patient. So we are basically university and the institute together. So we deal with the PhD students. We have the DM student, MCH surgery students, and 12 kind of the degrees we provide through the MCI, the Medical Council of India. It's a recognized all programs. So I'm just giving you the outset of the HCC. It's almost by 2025, it is 1 million cases worldwide, and the second leading cause of cancer-related deaths. And major etiologies are basically hepatitis B, C. It was, but nowadays it is more of the alcohol and more of the non-alcohol fatty liver diseases, because you know that almost all in the globe that the diet is changed and the people are having basically fatty liver diseases, which is now running that it is going to be HCC more. And that's the reason after controlling the hepatitis B and C, still we are on the NAFLD HCC patients. So what happens in the early stages, there is a chances of liver transplantation, resection or radioembolization. However, in the intermediate stage, when there is not only one tumor, there is more than tumor, there is again the radioembolization in the case. And in the advanced stage, when these two things are not working, we have of course, the drugs, sorafenib, which is making it for the HCC. However, what happens, there are high recurrence rates here, there is a chemo resistance here, and there is a survival increase by only 3 months with the sorafenib. So this problem stays as such if you treat or don't treat. So what happens during the normal liver, when you have the viral hepatitis, alcohol or non-alcohol fatty liver disease, so liver start giving the fibrosis. Fibrosis, it's a fibrotic. This stage is a fibrotic 1, 2, 3, 4. So last stage is a 3 and 4 considered as the cirrhosis. And this happens due to the telomere dysfunction, enhanced integrin signaling, and certainly leading to the HCC. So after the even treatment, there is a recurrence because there is a generation of cancer stem cells in that. And after even resection and chemotherapy, we believe that there are residual cancer stem cells and there are circulating tumor cells in this HCC left, which cause the recurrence and the metastasis. So what happens? What is the CTCs and CSCs similarities? I'm not talking about what they are, but what are their similarities? If you just take a general picture, where is the tumor microenvironment, there are the CTCs also, which is the epithelial based, and CSCs also, there are cancer stem cells. But they both, what they do, they invade into the capillaries and then they travel and then they go for the extravasations to the even the different organs also. So shared characteristics of CTCs and CSCs are that both play a critical role in cancer metastasis, spread of cancer from primary tumor to the distant site, highly resistant to the cancer treatments. Resistance is a major cause for the tumor recurrence and poor survival. However, there are overlapping markers, biomarkers, how you recognize the CTCs or cancer stem cells, that is also ACTAM or CD133, they are overlapping markers, and they both exhibit the properties of EMT, a process that allows cells to detach from original tumor and migrate. And of course, they have the ability to self-renewal. That's the reason they stay there together. So there is a diagnostic benefit. So we did the small portion of the patients. We did because it has a non-invasive sampling. You can take the blood sample, early detection of cancer and biological insight of cancer. Due to that, what we did, we took the few patients of the HCC and we found that can we see in the blood CTCs, and because those patients are random, like with the single lesion also, with the multiple lesions also, multifocal also. So we use the pan-CK and CD45 markers, and then this patient is with a single lesion, and this is with a multifocal, single lesion and multifocal, and we have observed that obviously in the CTCs in the multifocal patients is much more than with the single lesion. It means those CTCs with the multifocal lesion are breaking down from the tumor, and it is going to have the metastasis. So as I told you already, that CTCs and CSCs are the similar characteristics. It is very difficult to do the mechanistic studies only in the CTCs, so we took over the CSCs first. And our question, our clinician main problem is this: when HCC happens, so what? What we can do? We can't do anything. We can only change the duration of the patient. That good life pattern we can change. But to restrict the happening of the or generation of the tumor, we need to go before that. So before that is what stage? Cirrhosis. So there was no data that whether in the cirrhotic condition, how the hepatocyte, because cancer stem cell does not appear on their own. It is a transformation from the normal cells to the cancer stem cell, or normal hepatocyte change into the cancer stem cell. So there was a question, does cirrhosis, even fibrosis 3, 4 stages or even 2 stages, does that contains the cancer stem cells? So what we did in the similar study, this is Ritu Khosla, now she's a Dr. Ritu Khosla. So in her PhD thesis, we took the cirrhotic liver and we took the HCC, this is a tumor, but side by side the tumor, there is a portion which is non-tumorous, which we considered as the normal, because you can't take the normal liver from anybody. So, and this is of course, this is the best way that you take the normal, because it has the influence of tumor or not, that also you can see. So we took the cirrhotic patient, we took the HCC and normal, and as I told you, APCAM is one of the best marker along with the CD133. So we chosen as a APCAM positive cells. How? Because this is in the background that in the HG2 and HG7 cell lines, when we saw the expression of APCAM was around 70 to 80%. And when we have seen that primary human hepatocytes, their expression in APCAM was also more. So that becomes the first major biological marker which we can detect with the cancer stem cells. And we have seen that in the normal means this normal has the less APCAM expression, cirrhotic has the expression, and the HCC. Of course, cirrhosis has a lot of expression. And when in immunohistochemistry also, we have shown the similar data. What was the next? Does these APCAM, what we did, we took the tissue, we have made the single cell suspension, and then we did the flow cytometry. We found that the APCAM positive, we sorted those APCAM positive cells, and then we did that the stemless genes, and if you can see this APCAM blue is a APCAM normal stem cells, means normal patient, or normal tissue, sorry. CIR means cirrhosis, and HCC means HCC, or APCAM negative HCC also we took. When we are sorting, there is a negative and positive population, so we took the negative also to see whether that population also has. So you can see the yellow and the blue were not, but the cirrhotic and the HCC, they both had the stemless genes. So cirrhosis do express CSCs, that was the ultimate thing. And then these we checked whether these have after many signaling pathways, we found that there is a lot Wnt3 pathways running, and even with the beta-catenin, c-MEC, cyclin D1. So we found that this is very interesting, that the Wnt3, because now there is no mechanisms, connections or molecular pathways are still known for the cancer stem cells in tumor and in cirrhosis especially. So what we did, we inhibited the Wnt. Now as Swandon told me the 22 minutes, so I have to rush my language also and fasten the slides also. So you please bear with me. And what we did, we used the inhibitor, Wnt inhibitor. And as you can see, this is a spheroid assay. Here is a negative, no inhibitor. Here is an inhibitor. This is a normal stem cells. This is a cirrhotic and HCC. As we put the inhibitor, spheroids were less. So then we found it that spheroid goes away with the Wnt inhibitor. It means Wnt is one of the major, and we know that the HCC is driven by Wnt and the Notch pathways along with the hedgehog, certainly, but it is more. So now isolated ACTM positive, we did the transcriptomics, proteomics, transcriptomics, miRNA, and here also we took the same, and these are not the 1 even, it is almost 7, 8 patients, and then we found that the pathways, that if you can see, please pay the attention that there was a Wnt pathway was coming up very nicely, JNK pathway and Notch pathway. So because we started with the Wnt pathway and we blocked it and it was not happening, spheroid was going away. So we took it the Wnt pathway further, and then what we did, so I just little bit summarized here that what is happening, that in ACTM expression in the cirrhotic, just say C, and ACTM from the CSC means HCC, they both have the increased expression of ACTM positive. This ACTM positive is driven by the Wnt beta-catenin. Wnt beta-catenin gives the OX4 and analog and the SOX2 expression, which gives the increased stemness and increased proliferation with the c-MEC and the cyclin D1 I have shown you, and which causes the tumorigenicity. So in cirrhosis and HCC both has the ACTM positive, both have the ACTM positive CSC-like features, and microRNA and gene expression pattern of ACTM cirrhosis closely resemble to the ACTM positive CSC, means ACTM positive HCC. So now further, Ritu, what's there? That we did the proteomic and microRNA profiling. If you go through this table, the most one is the 10.64 is the ACTM CSC versus normal, and ACTM CSC versus HCC also. There is a 10.64, 5.43 fold HSP8. But when we have seen, when we combine the microRNA and proteomic profiling, HSP8 is regulated by MIR26, regulated by 20, but it is inhibiting. MIR26 5P is inhibiting. Then we have seen that what is the MIR? MIR26 5P, what is happening? It is combining with the ACTM also, and it is giving the with HSP8 also. So it means HSP8 is a central regulator which is giving the ACTM also, and which is regulating with by the MIR26B, because if you see MIR26B 5P is inhibiting the HSP8. So mechanistic story started from there, and we have observed that with this trio is happening with ACTM, HSP8 and the MIR26B 5P, and we have observed that the 26B 5P was minus 24 level. If you go back, so if you see that here HSA 593P is also there, which is regulating, which is inhibiting. But when we see observe the expression, it was more with the 26B 5P, whether it is a cirrhosis and this is a HCC. So we have observed that the RNA, relative microRNA expression in the ACTM CSC was very less when there was a mid-26B5P more. It means it is inhibiting, it is regulating the expression, and then there is a decreased mid-26B and increased HSP8. So it is a vice versa. It is going with the CSC and HCC more. So suppression of mid-26B5P enhanced the migration of HCC cells. What we did, we used the anti-mid-26, and we have seen within for 72 hours there was expression of this mid-26B relatively. When it expressed, it is giving the more of the HCC tumor, and it even that this cell closure assay shows that when you use the anti-mid-26B5P, there is a cell closure is more, and with anti-mid-26B5P, gap closure was completely more than the NC. So we know that ACTM as a target of mid-26B5P, and it directly also worked for the stemness through the Wnt, NANOG, OCT and SOX2. Then we have again used the assays of the using the anti-mid-26, that spheroids were increasing here and here. If you see that the both way, it means the anti-mid was the one pathway. But story does not end here, and story says that in HSP8 as a target of mid-26B, can we also downregulate the HSP8? And we have used the anti-mid-26B5P and seen that the HSP8 is increasing in this, but when we have used the mimic, it was decreasing. So it solves the problem, and it gives us the best that mid-26B5P inhibits apoptosis through HSP8 mediated degradation of BBC3. So we summarize here again that ACTM was more expression. It is giving the OX4, NANOG and SOX2. At the same time, mid-26B5P downregulation is increasing the ACTM and HSP8, which is giving the BBC3 through BBC3. We have the increased stemness, apoptosis and CSC maintenance. It means in the cirrhosis, we have the CLSC. Now the question comes, therapeutic strategies. What are the therapeutic strategies we can afford? Because we have the target of ACTM. So what we did, we got this kind of a short project from the DBT to develop the anti-ACTM aptamer equipped with the bacterial outer membrane vesicles. What we did, we took the bacterial outer membrane vesicles, we put the aptamer on the top of ACTM on OMVs and loaded with the doxorubicin, which is an anti-cancer drug, for their anti-tumor effect by specifically targeting the CSCs. So anti-ACTM doxorubicin loaded OMVs for treating HCC. So what we used the three aptamers, DNA aptamers 2, and one is the RNA DNA hybrid aptamer, and we have found that the docking capacity was certainly more in this 263, 256 and the 285. RNA DNA aptamer was the best aptamer with the cis5 on one side, which we can see fluorescence through the cells. And what we did it, first we found whether this ACTM aptamer is binding to the HG2 or HH7 cell line. We used the 50 nanomole, 100 nanomole and 150 nanomole, all these three aptamers, and we had found that the 89% with the RNA DNA binding is there here. And then what we did, we have found with the fluorescent microscopy that we found that again there was much more of the RNA DNA binding with the HG2. This is HG2 and this is HH7 cell line. And we thought that this is the best aptamer for the ACTM, anti-ACTM. And then what we did, that we have used the OMVs. These OMVs, our partner from the Netherlands, they were making it the bacterial OMVs. What happens that this Salmonellum typhimurium bacteria, it itself has the killing ability stability for the any tumor. But it has a toxicity also. So, but the OMVs, what they are producing, these OMVs are not toxic. So these OMVs plus DOX and plus we took the anti-FKM aptamer. So we used this as a target aptamer OMV DOX. We made the nude mice tumor and we did the IV injections, which showed us that the HCC shrinkage is there. We are still working on these OMVs. We are still working on that much more anti-aptamers, anti-FKM aptamers, we can use it, and OMVs if we can use it for the different metastatic tumors and in the mice model. So this is my acknowledgement. Of course, all the tumor samples come from the clinician. Professor Shiv Sarin is our director and the senior hepatologist. Gayatri Ramakrishna is my colleague, and Archana Rastogi is a pathologist. Ritu Khosla, who did almost all the work in this last 3-4 slides, Pushpa and other students also did. I have the national collaborators and the international, and very thankful to the funding agency from the India. I'm very thankful for the peaceful audience.
Any questions?
Yeah.
I think the amount of work in order to isolate the different samples from different patients, if it was done by one person, that's a lot of work. I don't know if
It's
that was.
pieces, and in India we give the pieces work more than 5 years to the student. So it's like this.
So it's about like 20 patients, or
It's only the 20 cirrhotic. It was more than 20 patients. HCC patient was more than 10, 12, and normal. Actually, every time you can't get even the normal liver also beside the tumor because the surgeon won't give you. The main question is that whether surgeon is giving you the tissue or not.
So I'm not too familiar with the FCAM based things, but FCAM is also expressed by epithelial cells onto a large extent, if I
Yes.
understand correctly. And so basically this drug would, the possible solution would be applied to the blood, normal blood pathway, but it doesn't migrate towards the epithelial surfaces at any group. How is this barrier formation kind of?
To conquer this problem, we took the normal because normal stem cells also express the ACTM. So ACTM was okay. It means that cancer stem cells also express, stem cells also express. So how you will define it? What we did, I have not shown, that when you in the nude mice make the NSCs and CSCs tumor, only CSCs were making the tumor, not the NSCs. And now this, our course is with the, not the only the subcutaneous tumor we generate, even we generate that in the liver, we generate the tumor with the CSCs. So because beneath the layer of the liver, the jelly, we put it the cells and we have seen the tumor over there also. So initially we were only doing, so it means that NSC don't have the, they have the expression of ACTAM, but they don't have the capacity to generate the tumor. So we have done it that, and that's the reason, that was the only reason that to go for the mechanism that how the ACTAM positive are driving the cancer stem cell maintenance and the proliferation. Because in cirrhosis there is a maintained cancer stem cells, it means. So you can't, so clinician worry was that you pinpoint before that, but it is already cancer stem cells, they are having it at the stage 3 and 4. So you, if you have to really nail down, you have to nail down before that, even fibrosis 1 and 2. Yeah, please.
One is this extracellular vesicles. So did you do the
It's not
characterization
extra
the OMVs?
Yeah.
These, did you do the characterization of them, because they may contain bacterial derived microRNAs, and nowadays we know that bacterial derived microRNAs can also target human environment.
Yeah.
So did you do a characterization of the contents
Yeah,
of
no.
the, because it's a pathogenic
Yeah. We have
bacteria.
not done it, but our partner did it in the Netherlands. They have published also, and they have even published that the full bacteria you take it, and then OMVs you take it. And even we have used, he has sent us both full bacteria also and OMVs also. So we used the full bacteria also. Even that was really lethal. That mice was not even surviving for 1 week. But OMVs went through it. We showed that there is reduction in the tumor was there with the OMVs.
So the question was like, why OMV suddenly? We could have taken, see, it's a anti-aptamer to EpCAM. I understand probably the aptamer is for targeting to the EpCAM positive cells.
EpCAM positive.
So we could have taken up some any other lipidic vesicle.
100%.
Yeah, but
100%.
what was the advantage of having the
Because this
Staphylococcus?
whole bacteria was quite targeting all the tumor cells. So we thought that these OMVs may be not having that much lethal, but OMVs as such have the specific, if we will target, and specific means killing capacity, then we can do it. But don't forget, it is a 2-year or 3-year back experiments. If you give me the now chance, I will put the more on extracellular vesicles and load it with the different drugs and use it. Certainly.
Last question. So since you use doxorubicin,
Yeah.
but in your earlier study you have shown catenin pathway to be the one which is
We put the
effective.
technique.
So if we have loaded the OMVs with a anti-catenin inhibitor or a Wnt inhibitor, then it would have been conjugated.
I am very thankful, and we thought about it, but yes, it's very, very good.
You can discuss.